to biology, the scientific study of life. Chemical communication. Many animals that communicate through odors emit chemical substances called pheromones. In most cases, both the production of pheromones and animal response to them are controlled genetically. Pheromones are especially common among mammals and insects and often relate to reproductive behaviors. Many moths, for example, emit pheromones that can attract mates from several kilometers away. Once the moths are together, pheromones also trigger specific courtship behavior. The context of a chemical signal can be as important as chemical itself. In a honeybee colony, pheromones produced by a queen and her daughters. The workers maintain the hive's very complex social order when male honeybee drones are outside the hive, where they can mate with a queen. They are attracted to her pheromone. When drones are inside the hive, they are unaffected by the queen's pheromone. Pheromones also function in non-reproductive behavior. For example, when a minnow or a catfish is injured, an alarm substance that is stored in glands in the fish's skin disappears in the water, inducing a fright response among other fish in the area. These nearby fish become more vigilant and group together in tightly packed schools, often near the bottom, where they are safe from attack. Pheromones can be very effective at low concentrations. For instance, just 1 centimeter cube for skin from a fathead minnow contains sufficient alarm substance induce an alarm reaction even when diluted by 550 8,000 liters of water. Auditory communication. The songs of most bird species are at least partly learned. By contrast, in many species of insects, mating rituals include characteristic songs that are generally under direct genetic control. In Drosophila species, males produce a song by vibrating their wings, and females are able to recognize the songs of males of their species through such details as in intervals between pulses of wing vibrations. The rhythm of the songs and the length of song pulses vary. Of evidence indicates that song structure in Drosophila males is controlled genetically and is under strong selective pressure. For instance, males reared in isolation produce a characteristic song for their species despite having no exposure to other singing males. In addition, the male song shows very little variation among individuals within a Drosophila species. While Drosophila species differ morphologically as well as in their courtship songs, some other insect species can be identified only through their courtship songs by behavior. For example, morphologically identical green lacewings found throughout Central to Northern Eurasia and North America were once thought to belong to a single species. Chrysoptera carnea. However, studies of their courtship songs by Charles Henry and his colleagues and students at University of Connecticut revealed the presence of at least 15 different species, each singing a different courtship song, as shown in this video. Several of these morphological identical species may occur together in the same habitat. An example of behavior is the isolation of species. Though they never observed hybrids of the different green lacewing species in the wild during more than 20 years of field work, Henry and his colleagues have been able to produce hybrids between different species under laboratory conditions. While all individuals in each species sing the same song, laboratory raised hybrid offspring sing songs that contain elements of the songs of both parental species. This data led the researchers to conclude that the various songs sung by the different green lacewing species are genetically controlled. Genetic influence on mating and parental behavior. Behavioral research has uncovered a variety of mammalian behavior that are under relatively strong genetic control, as well as the physiological mechanisms for these behaviors. One of the most striking lines of research concerns mating and parental behavior by male barrier voles, Microtus or Chirigaster. Prairie voles and a few other vole species, as shown this figure, are monogamous, a social trait found among only about 3% of mammalian species. Male prairie voles also help their mates care for young, another relatively uncommon trait among male mammals, in contrast with most other vole species. A male prairie vole forms a strong bear bond with a single female after they mate, associating closely with his mate and engaging in grooming and huddling behaviors, as shown this figure. Further, though an unmated male prairie vole shows little aggression toward other prairie voles, whether male or female. A mated male becomes intensely aggressive toward any strange male or female prairie voles, while remaining non-aggressive toward his mate. A few days following the birth of pups, a male prairie vole will spend a great deal of time hovering over them, licking them, and carrying them around while remaining vigilant for intruders. The genetic and physiological controls on the complex social and parental behaviors of prairie voles have been revealed over the past decades by the research of Thomas Insel and his colleagues at Emory University. Early research suggested that arginine vasopressin AVP, a 9 amino acid neurotransmitter released during mating, might mediate both pre-bond formation and aggression by male prairie voles. In the central nervous system, AVP binds with receptor called the V1A receptor. The Emory researchers found significant differences between the distribution of V1A receptors in the brains of monogamous prairie voles and their distribution in Bromicus montans voles, which live in the mountainous regions of western North America. To test whether the distribution of V1A receptors is a key factor controlling the mating and parental behavior of prairie voles, Insel and his colleagues inserted the prairie vole VA1 or 1A receptor gene into laboratory mice. Not only did the transgenic mice develop brains in which the distribution of VA1 receptors was remarkably similar to the distribution found in prairie voles, but they also showed many of the same mating behaviors as monogamous male prairie voles. In contrast, male wild-type mice did not have the same V1A receptor distribution and did not show such behavior. Thus, though many genes influence the mating behavior of these voles, it appears that a single gene may mediate a considerable amount of complex mating behavior and perhaps parental behavior. The cures of prairie voles, it remains to be seen whether other complex behaviors are under similarly simple genetic influence. In addition, although genes influence behavior in a multitude of ways, environment also has major effects on behaviors, as we shall see in the